The overall goal of this procedure is to image and quantify the number of 19F labeled cells in vivo using 19F MRI. This can be done in a clinical or preclinical setting. This is accomplished by first labeling cells with a suitable 19F agent. This is most often done ex vivo. The second step is to quantify the 19F content per cell, which can be done using 19F-NMR with a known reference. The 19F content per cell is assumed to remain constant during the experiment, otherwise, correction factors must be calculated. Next, the subject is imaged in vivo using 1H and 19F MRI to localize the transferred labeled cells. A known reference and appropriate imaging parameters are essential for quantification from the image data. The final step is to process the image data to calculate the number of cells. Ex vivo histology or other techniques can be used to corroborate the data where possible. Ultimately, 19F MRI for in vivo cell tracking can be used to study the transferred cells in terms of their localization and numbers over the relevant length of time, given certain assumptions as outlined in the text.
The main advantage of this technique over existing methods, like 1H MRI using superparamagnetic cell labels, is that there's no confounding background signal and that the number of cells can be determined directly from the image data.
This method can help answer key questions in the field of cellular therapeutics, such as where are the cells and how many are present at various time points.